Next up is Peter Cousins, who's had a long and storied history in the lab, and then went to the USDA in Geneva and worked as a rootstock breeder, and now has come back and to work on the dark side, the other dark side, side of the room. He's now the grape breeder at Gallo, so he'll give you some idea of how things are going there too.
Thanks, Andy. Yeah, my mom tells me that imitation is the most sincere form of flattery, and I'm a grape breeder too. So thank you for showing me the way. And thanks for our other speakers. We're talking about the future. I'm not going to talk about what I did. I'm going to talk about what's coming to you. This is a talk about opportunity. Nobody is going to come to your vineyard and pull up your vines and tell you what to do. A grape breeder is intended to make your life more fulfilling, to provide opportunity for you. It will not make the future more simple. It will provide you with more choices. All right? So I'm here to tell you that things are going to become more complicated. Let's talk about clones. Clones in grapevine are vegetatively propagated lineages. They do not have an implied message of uniformity or difference or quality. So you may have a special clone that came off of your grandmother's fence, and it's important to you because of where it came from. That kind of clone is not what we're talking about. We're talking about clones which provide differences among the plant materials which are important in viticulture enology. Now, UC Davis, and particularly Foundation Plant Services, the Department of Viticulture Enology, and Plant Pathology have really paved the way for distinguishing the kinds of differences among clones which are important. Right now, those kinds of differences are driven by two main areas. One of them are differences in pathogen infection, particularly viruses. The other kind are differences in genetics. So first, let's talk about the virus differences. Because of work done here on campus, the ability to detect viruses in plant material is better and faster than ever before, and it's getting better and faster. Our ability to eliminate the viruses, that is, to provide plant material which tests free from viruses, is getting better and faster also. Projection. In the future, all plant material will leave the nursery testing free from virus. It will become to the point where we can actually test every individual plant because the cost will be so low. And that plant material will have been sourced from Foundation Plant Materials or another location where the original source material is tested free from virus. All right, so we're going to preserve our ability in a legal sense, regulatory, to plant whatever filthy plant material you'd like, but it will not be the responsibility from a nursery which has accidentally contributed plant material to you that is infected with virus. We'll have the opportunity to have 100% virus-free plant material leaving the nursery welcomed into your vineyard. Let's talk about the genetic differences. Right now, there are very few clones which are truly distinguished by a known genetic difference which actually drives a performance difference among the clones. There are a few. For example, we know that the aromatic clone of Chardonnay, Chardonnay Muscat, differs from Chardonnay clone 4 because of a specific DNA change. That base pair difference is known and it can be identified by sequencing. So that's an involved process, but we know that the difference in clone, which gives you a difference in performance in the vineyard, is due to a specific genetic difference. In the future, we will be planting clones which are different because of genetics. We'll have eliminated all the differences which are due to viruses. You can still keep the ones which came from your grandmother's patio. But we will know that the differences among the clones are due to a specific genetic difference, and we'll know what the drivers are. Not just a fingerprint, but what the actual sequence or genetic difference which is driving that, and that's very important. Hans showed us some differences among clones of vignoles in cluster compactness. And at this point, we don't know what the differences are genetically, which makes that happen. We know that there's a performance difference, and through detailed genetic sequencing, we'll be able to identify the specific genetic changes which are responsible. We saw the information from Samuel about the differences in the ripening time and other aspects of the phenology of Cabernet Sauvignon. We know that in many varieties, for example, in Pinot Noir, in Flame Seedless, in Concord, there are early ripening sports or clones, and we'll know in the future the specific genetic differences which are responsible for that useful genetic change, which gives you a difference in performance in the vineyard, right? So we'll still have clones in the future because people like to drink Chardonnay, we like to drink Cabernet Sauvignon. Those have a huge footprint in the industry, and new varieties will be introduced by breeding, but then of course we'll also have clones within those varieties as well. Technology is coming to help us. Well, we've seen these new varieties that Andy developed, and a special call out for the delivery of new plant material which has useful disease resistance as well as demonstrated enological properties and wine quality. So, with regard to Pierce's disease, this disease has been with us and was first identified in Southern California by Newton Pierce. They thought at first it was a virus, so it's not a virus. There are grapevines which coexist with Pierce's disease in nature, and there were varieties which were developed which could be grown in those places like in Florida, where Pierce's disease is an even bigger problem than in California. But the plant material that Andy identified and used in breeding is particularly useful because it has true biological resistance against the Pierce's disease and the bacteria which cause that disease. So, existing problem, new germplasm, new approaches. How does that apply to other kinds of diseases, not just to Pierce's disease and for other parts of the grape and wine community? We know that powdery mildew is a huge problem, and it's a North American pathogen which has now spread around the world. Today, we can buy powdery mildew resistant wine grapes, and those were developed by breeding, particularly new varieties coming to us from France, from Italy, from Germany. For example, Ardipon, Sorelli, and Collardus Muscat. These are quality wine varieties. They are not the same as Chardonnay. They're not the same as Cabernet Sauvignon. But they have been developed not only looking at the pathogen side, the pest resistance, but also with an eye to quality. Well, you may say, gosh, I've had Foch. How are these new resistant varieties better? Isn't the source of resistance the same? What could we possibly be motivating us to plant these new kinds? The answer is that we do have new sources of resistance, and Andy has really shown this in his work with the Pierce's disease, but we know that in the case of powdery mildew, he's also identified resistance sources coming from Vitis piazetzkii, which is a wild grape that's native to China, and worked with resistance coming from other types to identify molecular markers and conduct a breeding to introduce the resistance into a quality background. Small-scale research winemaking enables us to evaluate not just the viticultural performance, but the enological properties of these new selections, and that's really very essential. If you're not evaluating how these grapes turn into wine, both from the production side and also the sensory aspects, then we're just breeding grapes, we're not breeding wine grapes. And we can see that the opportunity to reduce our pesticide application while continuing to produce quality wine, that revolution is coming to California. Nobody's coming to tell you what to do with your clones, but the regulatory environment worldwide is changing, right? So in Europe, there's an increased emphasis on breeding for disease resistance because of an EU approach to reducing pesticide applications in all agriculture, and specifically grapes are a huge part of that footprint, and the instructions are very clear. You need to reduce the amount of pesticides being used. And we still want to continue to make quality wine. So the breeding programs in Europe are ramping up again, as they haven't for decades, in order to bring small-scale research winemaking grape and wine composition studies, and disease resistance, along with the use of DNA molecular markers to predict performance and disease resistance to deliver those new varieties. In France, they have regional breeding programs so that the grapes which are being developed, which are resistant to downy and powdery mildew for southern France, have different attributes than the ones being developed for Bordeaux or for other quality wine regions, which we are all familiar with. More than a dozen regional programs in order to have varieties which are suited, adapted, resistant, and enthusiastically accepted. So we're going to see new varieties coming that build on this theme. We already knew that we had the resistance, we already knew that we could have quality, and we're going to bring those together. Rootstock breeding will never be the same again. There's genetic resistance to fan leaf, and it comes to us from Riesling. Part of Andy's graduate work was identifying genetic resistance to fan leaf, which came from vinifera. And now that work is demonstrated to come to us from Riesling. That Riesling is genetically resistant to the fan leaf virus when it's introduced to the plant through the roots. That means that we have the opportunity through plant material to eliminate fan leaf virus forever through the use of new plant material. It's something like if we breed new varieties, we're going to have fan leaf resistance, which comes to us from Riesling, and we're going to have nematode resistance, Xiphidium index resistance, probably coming to us from Vitis arizonica. Thanks for the molecular markers and for doing the pioneering work in that area. And we'll have to have phylloxera resistance. Of course, Andy and his team, and Carl, talked about the evaluation of the resistance and the populations. Against the phylloxera, we know that there are very strong sources for phylloxera resistance, and those sources need to be combined. We can really produce phylloxera-resistant, fan leaf-resistant rootstocks. And with the use of that type of plant material, because fan leaf is exclusively a grapevine pest or virus, we can really eliminate that forever by the use of resistant plant material. Fan leaf is a serious problem when you have it. It's patchy. The nematode doesn't get around very well. I'm sure Glenn never distributed any when you were busy distributing phylloxera. But there are other viruses which get around a little bit faster, one of them being those in the leaf roll complex and those related viruses which can be spread by mealybugs. We use this as an example for what the future promises because there is genetic resistance in grapevine germplasm against the important mealybug species. And we know that the use of this plant material gives you a very strong reduction in the mealybug numbers. The literature suggests to us that there is resistance, genetic resistance against leaf roll as well. The delivery of new plant material, which has these attributes, is a long-term proposition, so bear with us. But we know that there are sources of resistance identified for many of the important pathogens, for example, crown gall disease, and Samuel, you showed us the Margaroides, and there may yet be resistance against those as well. So we look forward to the revolution in plant material. It does not happen overnight. Thank you, Andy, for leading the way. Thank you.